So now we've completed the first section of this presentation. We're going to move to the second section of the presentation. For the second section, I've now illustrated or described for you an understanding of the homing cascade and the biology of cell migration. We're now going to go into details on the molecular effectors of homing to bone marrow. Homing to marrow is an active process. As a bone marrow transplant physician, this was the principal objective of my research for the first 15 years that I was in the field. I sought to determine what molecules were regulating the trafficking of the hematopoietic stem cells I was introducing into my patients. When we do a bone marrow transplant, the hematopoietic stem cells are not injected back into the marrow, they're injected into the bloodstream, usually through central vein access in the jugular or the subclavian vein, and the cells will then traffic to the bone marrow. I wanted to understand the molecular basis of that process. Again, this process is an active process where the cells will be introduced in the bloodstream and will literally be recruited into the marrow microenvironment. So from work in our laboratory and the work of other laboratories, a variety of different molecules were identified that were critical for this process. Importantly, at the first step in the process, we identified that E-selectin, a molecule which is normally inducibly expressed, and endothelial selectin, which I'll tell you more about in just a second, was permanently expressed on the marrow microvascular endothelial cells, and that hematopoietic stem cells were first coming down and finding endothelial beds in the marrow because they expressed E-selectin ligands. The cells would then tether and roll on that E-selectin, and then they would be engaging a chemokine known as SDF1, otherwise known as CXCL12, which is constitutively expressed in the bone marrow microenvironment, which through contact with the chemokine receptor CXCR4, which is present on the hematopoietic stem cell, would then upregulate VLA4 integrin, VLA4, a beta-1 integrin, binding to its ligand VCAM1. And then the cell would firmly adhere and transmigrate into the marrow. These were steps that our laboratory and other laboratories had worked out. Just to review here then, the roles of E-selectin and SDF1, otherwise known as CXCL12, in this homing process. Well, first it was determined that E-selectin is permanently expressed on marrow sinusoidal endothelial cells. That the chemokine CXCL12, that is SDF1, is the principal chemokine which directs the hematopoietic stem cell migration to the marrow. And the receptor for this molecule is CXCR4. Hematopoietic stem cells express high levels of functional CXCR4. So that the SDF1 molecule, the CXCL12 molecule, which is constitutively expressed in the marrow, then engages the CXCR4 on the hematopoietic stem cell surface, upregulating VLA4 activation and firm adherence on VCAM1. That VCAM1 molecule is also expressed constitutively in the marrow endothelial beds. Importantly, however, SDF1 expression is markedly upregulated at all sites of inflammation. So though it is permanently expressed in the marrow, it is upregulated in all tissue beds at all sites of inflammation. And also E-selectin is upregulated at all sites of inflammation. So right there you can see where a molecular drill played out in the marrow would be important to deriving cellular trafficking to sites of inflammation. Now what about this molecule E-selectin? E-selectin is an endothelial selectin. It's typically an inducible molecule induced by inflammatory cytokines TNF-alpha and IL-1, which forces it to be highly expressed at all sites of tissue injury. It literally functions to capture cells under blood flow conditions as it passes by that endothelial bed. However, E-selectin is constitutively, that is permanently expressed, on marrow microvessels and also dermal, that is skin microvessels. Now E-selectin is a lectin. By definition, a lectin is a protein that binds carbohydrate. So therefore, this structure is looking for a sugar motif. The critical sugar motif that it binds to is a tetrasaccharide, a 4-sugar composition called sialylated Lewis X, or its stereoisomer called sialyl-Lewis A. All E-selectin ligands contain this 4-sugar motif, this either sialylated Lewis X or isomer sialylated Lewis A motif. Now the selectins are a family of 3 lectins, E-selectin being expressed on the endothelial beds, P-selectin expressed on platelets and also on endothelial cells, and L-selectin, which is only expressed on leukocytes or white cell series. Importantly, L-selectin is also expressed on hematopoietic stem cells. All 3 of these molecules forming this family, called the selectin family, are calcium-dependent lectins. That is, they require calcium for binding to their sugar motifs. They mediate cell tethering and rolling interactions, and they are the principal effectors of the step 1 interactions. So these molecules and their ligands are the best homing receptors. L-selectin, as I said, is found constitutively on leukocytes and hematopoietic progenitors. E-selectin on stimulated endothelium, that's why it's called E, because it's only found on endothelial cells. And P-selectin is on platelets and also endothelium. The P-selectin on platelets is inducible, but in the mouse, not in the human, in the mouse, P-selectin is also constitutively expressed in the marrow microvascular endothelial cells. Now, a critical difference between humans and all, I should say primates and all other species needs to be emphasized here. The promoter, which induces E-selectin expression, the TNF-alpha and IL-1 responsive promoter, which induces E-selectin expression, is uniquely retained in primates. That same promoter is present on the P-selectin gene in all other species. So that in a mouse, P-selectin dominates in recruitment of cells to sites of inflammation. But in humans, P-selectin does not function. It is only E-selectin that functions in the recruitment phase to an inflammatory site. So you may have upregulated P-selectin expression at endothelial beds because it is preformed in the Weibel-Palade bodies of the endothelium and in the alpha granules of the platelets, and it can be translocated to the membrane to transiently be upregulated at sites of inflammation. But operationally, the E-selectin gene gets massively turned on within 2 to 4 hours, and only E-selectin thereafter dominates to recruit cells in primates, that is, human beings. So there's a big difference in biology of mouse studies and human biologies in human regarding the selectin function. So the canonical sugar motif that selectins bind to, all of them, and most importantly E-selectin, is this 4-sugar structure, here depicted as sialylated Lewis X, and on the right, the isomer sialylated Lewis A. In each case, there are a combination of 4 sugars, a sialic acid, galactose, an N-acetylglucosamine, and a fucose. And the linkages differ in that in sialylated Lewis X, you have a type 2 lactosamine backbone. By definition, lactosamine is a galactose linked to N-acetylglucosamine. And that's a beta-1,4 linkage in a type 2 lactosamine, and a type 1 lactosamine, it's a beta-1,3. In that case, the fucose here that's attached to the N-acetylglucosamine is in an alpha-1,4 conformation in sialylated Lewis A and an alpha-1,3 conformation in sialylated Lewis X. And in each case, the sialic acid is linked to galactose in an alpha-2,3 conformation. Now, importantly, these motifs can be expressed on O or N glycans of a core protein and can also be expressed on glycolipids. So glycolipids can function as ligands for E, L, and P-selectin. Sialylated Lewis X and its stereoisomer sialylated Lewis A are each recognized by the monoclonal antibody HECA452, which makes that monoclonal a useful reagent to identify potential selectin ligands. So, shown here in cartoon fashion is a cell in blood flow which possesses the relevant tetrasaccharide sequences sialylated Lewis X or sialylated Lewis A. And if there's E-selectin on that endothelial bed, that endothelial lining, those cells will then tether and roll on that endothelial cell. So by virtue of expressing this tetrasaccharide sequence in the cell in flow, and by virtue of the endothelial bed displaying this particular selectin, E-selectin, a cell will begin the tethering and rolling actions which are prerequisite and fundamental to trafficking of that cell into a tissue bed. And in each case, the sialylated Lewis X or A glycans can be recognized by monoclonal antibody HECA452. So our laboratory goal from 1987 to 2000 was to find that bone marrow homing receptor, that is, the most potent E-selectin ligand expressed on hematopoietic stem cells. And all our biochemical studies over that period of time ultimately led us to identify that molecule, and that molecule is called hematopoietic cell E and L-selectin ligand, as shown in abbreviated form H-cell. H-cell is a sialylated fucosylated glycoform of a well-known molecule, CD44. CD44 is ubiquitously expressed on essentially every cell of the body, but only on hematopoietic stem cells. Importantly, only on hematopoietic stem cells. The CD44 core protein scaffold gets decorated with sialylated Lewis X, such that that molecule is then H-cell. And that H-cell molecule is the most potent E-selectin ligand and directs the trafficking of hematopoietic stem cells in the human to human bone marrow endothelial cells. Now, an important distinction has to be made here as well. Mouse hematopoietic stem cells do not express H-cell. So whatever parallel studies are being performed in the mouse related to the homing of hematopoietic stem cells to the marrow are non-relevant or irrelevant to the condition in a human. In the human, the molecule that mediates this process is H-cell. So let's talk a little bit about CD44. Importantly, CD44, the gene itself, contains 9 splice exons in a human and 10 splice exons in a mouse. It's beyond our time today to discuss the polymorphic variants of CD44, except to bring out the point that the CD44 expressed on hematopoietic stem cells is the, quote, standard form of CD44, which lacks all splice variants. So its core molecular weight is approximately 35,000. However, it's glycosylated normally to about 90 to 100,000 molecular weight. And then a subset of CD44, only present on hematopoietic stem cells, gets the sialylated Lewis X motif on it to convert it to H-cell. It is a remarkably diverse and remarkably functional, multifunctional molecule, and is present on all adult stem cells, and it's involved in important biological functions, speaking of CD44, such as cell adhesion, cell migration, cell differentiation, and signal transduction. And it is the principal receptor for hyaluronic acid, and it's best known as the hyaluronic acid receptor. H-cell, on the other hand, is a sialylated fucosylated, that is S-lex, bearing glycoform of human CD44 expressed on the earliest hematopoietic progenitor cells. Those are usually defined by surface marker CD34 and the absence of the marker CD38 and the absence of any lineage differentiation markers on the cells. So that is the CD34 positive, 38 negative, Lin negative population of hematopoietic stem cells. It's defined on hematopoietic stem cells, HSC, as CD44, which is reactive to monoclonal antibody HECA452 and another anti-sialylated Lewis X monoclonal antibody known as KM93. It can also be identified by binding to an E-selectin immunoglobulin fusion protein, EIG, which functions essentially like an antibody in locating E-selectin ligands. So as CD44, if you do a Western blot immunoprecipitating the CD44 molecule and it's reactive with HECA452 or E-selectin IG chimera, it is by definition H-cell. It is the most potent E-selectin ligand expressed natively on any human cell. I want to emphasize that H-cell is not CD44. H-cell is sialylated Lewis X plus CD44. By the same token, CD44 is not a selectin ligand. It is inappropriate to claim that CD44 is a selectin ligand because CD44 has multiple pleiotropic forms, polymorphic forms with a lot of different functions, and only a very small subset of the CD44 molecule is decorated with the relevant carbohydrate to allow it to bind to E-selectin or L-selectin. When that CD44 is decorated with a sialylated Lewis X motif, it is then H-cell. The working end of CD44 in binding hyaluronic acid is the protein itself. The deglycosylated CD44 binds hyaluronic acid better, in fact, than a glycosylated CD44 molecule. However, on the other hand, the glycans, the carbohydrate modifications of CD44, confer the selectin binding capability. Indeed, you can denature the core CD44 protein completely, and the 8-cell function is intact. So therefore, the carbohydrate provides the function to the CD44 scaffold. That's why it's so important to emphasize that molecule is 8-cell. So if we go through the molecular components of cellular homing to marrow now, I want to point out the critical role of 8-cell in mediating the binding to E-selectin, and the role again of CXCL12 in recognizing SDF, but the also important role of the beta-1 integrin at the tail end in the step 3 activation and transmigration process and binding to its ligand VCAM1. Now, if you look at the homing receptors, of all of the homing receptor family that's expressed on hematopoietic stem cells, you'll see that there's a redundancy of homing receptors expressed on hematopoietic stem cells. HSCs express L-selectin, CD44 itself, and 8-cell, the glycosylated variant. They express PSGL1 and the glycosylated variant that binds E-selectin, the CLA molecule. They express CD43, they express alpha-4 beta-7, they express VLA4 and LFA1. However, if you look at mesenchymal stem cells, they only express 2 homing receptors, and 1 out of the 2, as I've mentioned before, the VLA4 molecule, is a very poor homing receptor. It needs to be activated for it to function as a molecular brake. The other one is CD44 that can bind hyaluronic acid. However, it lacks all of these other homing receptors. Importantly, it lacks E-selectin ligands. So, just in review, mesenchymal stem cells lack ligands for E-selectin. They can't dock on the E-selectin molecule. They do not natively express 8-cell, but express large amounts of the CD44 molecule. This led us to ask a simple question. Could we glycan engineer the CD44 molecule on the mesenchymal stem cell surface itself to create the 8-cell molecule? And if we were capable of that glycan engineering, would those live functional mesenchymal stem cells traffic into the marrow and make bone in the marrow? That was the question.